We have the ability to shape ourselves by way of the choices we make. Yet much of who or what we are is determined far before our arrival, embedded in a sequence of inherited traits that, ideally, contain our ancestors' greatest strengths. This bedrock of biology defines our genetic code. But what if I told you there was a way to crack that code?
Sorry, what is this for again?
Our overlords demanded that you be vaporized, but we like you the way you are. So we're meddling with your genetics just enough to destroy everything you once were. That way we can tell HQ the truth while simultaneously keeping you in our lives.
Ah, like dyeing your clothes a different color.
Can't we just dye my hair then?
It's a bit more complicated than that. Right, Herb?
There are a myriad of ethical and moral issues that come with the ability to manipulate the human genome, turning DNA into D&D, min-maxing our genetic makeup. But for our needs today, we'll be using a magical metal MacGuffin that warps reality.
I found the right program, but it won't let me access it. I even tried executing it as an admin.
Speaking of executed admins, I better take this call.
DNA, or deoxyribonucleic acid, is like a big, long piece of code that determines everything about us. How tall we are, our sex, and yes, dunk, even our tolerance of dairy.
I always thought I was cursed by a cheese witch who made it so my body hurt every time I ate a cheese witch.
DNA is organized into a double helix. A fancy way of saying it looks like a twisting ladder. The rails are made up of sugars with phosphate groups, and each rung is a pair of complementary nitrogenous bases. In DNA, these bases could be adenine, cytosine, guanine, or thymine. They interlock, so A pairs with T, and C pairs with G. When a cell needs to make something, a section of this ladder is temporarily split down the middle. And you end up with two single DNA strands, each with their own sugar-phosphate backbones and exposed unpaired bases.
Thank you for cutting my ladder in half. It's actually super easy for me to find a new ladder at the bottom of the ocean. Explain to me, how does this end up working as a code?
One strand is used as a template to make RNA. Free bases pair up with the newly exposed DNA section and create a complementary RNA copy of the gene. A notable difference is that RNA pairs adenine with uracil, not thymine, and RNA's sugar-phosphate backbone is slightly different. Once finished, the messenger RNA leaves the nucleus to find a ribosome to read it, and the DNA is zipped back up. In sets of 3 nucleotides called codons, RNA is translated into amino acids and begins folding into proteins. These proteins are the final product that starts doing things inside of us. If we think of each nucleotide as a letter, codons could be words, and DNA is the full instructions for life. Sections of DNA are called genes, and they encode specific information, and the entire structure is called the genome. We actually only finished mapping the entire human genome back in April 2003. It was done by the Human Genome Project, or HGP, which started back in 1990. The HGP also mapped out genomes of several other creatures, including the bacterium E. coli, baker's yeast, fruit flies, nematodes, and mice. Since then, we've mapped out even more animal genomes, including bear's dung.
I don't know how that would be relevant to me, but okay.
We've even mapped out the DNA of our primitive Neanderthal cousins back in 2010, which allowed scientists and archaeologists to discover that many people alive today have Neanderthal genes. Personally, I think Big J is 100% Neanderthal, but I digress.
Okay, sure. It doesn't even begin to help me change my DNA.
It is technically possible, but we're talking about going into DNA and changing specific sections to control what information is being encoded. We certainly don't have the equipment on board to do that.
Equipment, you say? If the machine can't edit me, I'll have to edit the machine! Yow!
He's changing into somebody else! Someone who has been electrocuted!
In an effort to abandon my birthright, I dabbled with the idea of manipulating my own DNA. I ended up just running away from home instead. Fortunately, there is another way to edit DNA using a different technology. Does the name CRISPR mean anything to you?
Like these chips?
They're baked crackers.
I mean clustered, regularly interspaced short palindromic repeats, or CRISPR. It's a bacterial immune system that scientists Dr. Jennifer Doudna of UC Berkeley and Dr. Emmanuel Charpentier of the Max Planck Unit for the Science of Pathogen discovered. They even won a Nobel Prize in chemistry for their breakthroughs, and they were the first all-women team to win one. Hashtag women in STEM, am I right?
Did you forget that HQ Ocean gated my empty shuttle? Yeah, feminism can wait.
Whoa!
Not cool, man.
It sounds like you're a performative male.
I am sorry, but yeah, no, definitely tell me how CRISPR works.
It works in two parts. The first part is a CRISPR-associated Cas nuclease. Nucleases are enzymes that break down nucleic acids like DNA. And the second part is a guide RNA. Together, they create a double-stranded break at a specific spot, opening it up for gene editing goodness. Once the Cas nuclease cuts the cell's DNA, its natural repair system kicks in. This usually leads to missing bases and a non-functional gene. This is what's called knocking out a gene.
I have a pair of non-functional genes. They're too dang small.
Dunk. All skinny genes are non-functional. If you want to add a new gene or change a gene to fix it, you need to add a new piece of template DNA with the sequence you want. This is called knocking in a gene. Unfortunately for you, it's not super practical to edit adult DNA. Adult genes are already expressed and expressing given certain conditions. We would have to edit an astronomical number of cells. This isn't to say that we can't use CRISPR to affect adult genes. Victoria Gray, a 37-year-old woman from Mississippi, suffered from sickle cell disease, and she experienced chronic pain and fatigue throughout her life. She said, "The pain I would feel in my body was like being struck by lightning and hit by a freight train all at once." Sickle cell disease affects hemoglobin in red blood cells, and after an experimental treatment using CRISPR-Cas9 in 2019, doctors were able to express a gene usually only found in fetuses supporting her body's production of hemoglobin.
So it does work on adults!
In some very specific circumstances, yes. This is all very new technology, so scientists are still learning what we're able to do with it. It's far more practical to edit the genes of a pre-developed embryo.
Editing your baby?
This is the dark shadow looming over the whole breakthrough. In 2018, Chinese researcher He Jiankui forged documents and lied to doctors to implant gene-edited embryos into 2 women without their consent. Jiankui announced a plan to edit the genes of several human embryos to give them resistance against HIV, which would hopefully be passed on to their descendants as well. He hoped the research could be used to help fight against HIV and AIDS in Africa. His research was banned after critics argued there were better and safer ways to fight HIV infections, and that gene editing the embryos would potentially expose the babies to side effects that just weren't worth the benefit. Despite this, he went ahead and did it anyway.
That is messed up. Were his intentions good at all?
To quote Jurassic Park 3, my favorite movie, some of the worst things imaginable have been done with the best intentions. As some folks might know, it's common practice to screen in vitro fertilized embryos for genetic diseases that might complicate pregnancy or impact a child's quality of life. This screening also allows parents to choose the sex of their baby simply by choosing only to implant the embryos with the sex of their choice. This isn't gene editing, though.
Discarding embryos with the wrong sex seems, dubious at least.
It's one of the most controversial aspects of IVF. Most people would agree that it's not ideal to bring a baby into the world you know will have a genetic disease that will affect their quality of life.
Everyone's definition for a quality life is different and has more factors than just genetic ones.
Bioethicist Vardit Ravitsky, in an interview with Associate Editor Molly McDonough with Harvard Medicine, said that for the vast majority of human existence, we've had no access to information about pregnancy. We knew the outcome, you have a baby. But that's about it. Even access to ultrasound is wizard-level knowledge compared to the rest of human history. Ravitsky says that any type of program meant to give people information prenatally must have the objective of enhancing reproductive autonomy. She continues, saying that eliminating certain conditions, traits, syndromes, and types of people should not be the goal of this kind of genetic screening. Ravitsky also notes that physicians need to be careful about their bias when they give potential parents this information. But some individuals with loose relationships to ethics have their eye on a more, how should I put it, designer goal. Using tools like CRISPR, you could hypothetically edit your baby so it's taller when it grows up, change their eye color, modify their metabolism. If genetic screening during IVF can create ethical problems, then gene editing your tailor-made designer handbag baby is a full-send adoption of eugenics. For the blissfully unaware, this is the philosophy and act of manipulating human genetics to reinforce traits we find desirable. It has historically almost always been used to maintain systems of racism, ableism, and anti-Semitism. See Germany, circa 1930. The UN has made statements disavowing the use of gene editing for anything other than preventative, diagnostic, or therapeutic reasons.
Interesting stuff. Yeah, all of that was really good. So to clarify, none of this information is going to help me, right?
No.
What do you mean? Why'd we even talk about all this stuff?
I had thought expounding on gene editing would lead to a plot beat where we find closure. That's how this format works, right?
Deal with an issue, riff on a topic related to it, and then antics ensue?
I don't think that's going to happen today.
Yeah, no, it's fine. It's okay. Really, you did your best.
Don't panic, Big J. We'll figure this out.
You know, it's cool, Dunk. I don't have a choice but to be good with it, so I'm good.
Oh, gods! It's all coming to an end!
Oh no, what's happening? I'm matching your vibe.
They're here, Dunk! HQ!
Wait,
They're here,
HQ's here?
Dunk!
No, I have to... My browser. My browser.
Big J, my precious boy, I'm so sorry.
No, my family! I just found them!
Hey, this is Big J. You might have heard we have a Patreon. We definitely don't have much on it. Hey, and if you don't want to do that, maybe you can just like, comment, and subscribe. That would be just as swell. Maybe the splash will not be as big, but the splash will be definitely noticeable. I haven't done one of these yet. This is my first one. That's it. The circle's